Good morning, everyone.
A very warm welcome to the second day of SIMPIC and to all of you joining us for today's conference session.
A special welcome to visiting professors, medical students, and high schoolers who have made the effort to be here. It is a wonderful thing to see such a diverse group gathered here in this room.
While our delegates are currently taking part in the first round of the competition, this conference serves as another platform for learning, discussion, and inspiration.
Before we begin, allow us to introduce ourselves. My name is Nanapat Tamuntri.
And my name is Viran Patiranpruk. We are truly delighted to serve as your Masters of Ceremonies for today's conference and will be guiding you through today's program and comprehensive speaker lineup.
This morning's conference will be running from 9 a.m. to 12 p.m. The program will be divided into 4 sections, each led by leading experts in the field.
The sections are designed to offer insights into important topics that are not only relevant to the competition, but to the future trend of medicine and healthcare.
The first section, starting from 9:10 to 9:35 a.m., will be delivered by Professor Sid Sathon Sumeti, Deputy Dean of Research and Innovation, who will introduce you to the fascinating world of research and development in SI Research Inspiration.
Following that, from 9:35 to 10:35 a.m., we will explore a topic that could potentially redefine the way we understand aging, with Professor Emeritus Dr. Siriraj Songsivilai on Forever Young, why microbiology and immunology will help define human longevity.
Wow, that sounds like an intriguing topic. Having explored those broader aspects of research, we will then shift our focus to bridging clinical needs with research, lessons learned from neglected parasitic diseases. Delivered by Assistant Professor Dr. Pachalapon Desha-Sintana Salasombat from the Parasitology Department from 10:15 to 10:55 a.m.
And finally, from 10:55 to 11:45 a.m., Dr. Janjira Thaipadungpanit from the Faculty of Tropical Medicine will show us how a microbiology technique is used to track and stop a disease before it can spread in genomic tracking of the human leptospirosis epidemic in Thailand.
Each of the sections offer a unique and insightful perspective, and we are certain that this morning will be nothing short of inspirational for us all.
Please note that the last 10 minutes of the last 3 sections will be a Q&A session where we strongly encourage all participants to take part.
It is a valuable opportunity to engage directly with our distinguished speakers and deepen your knowledge and understanding of this subject matter.
Before we begin, may we kindly ask that your devices are set to silent mode, and for your convenience, the toilets are accessible via the side exits.
With that, let's begin our first session. It is our privilege to welcome Professor Sid Sathon Sumeti, who will introduce us to Siriraj's research inspiration.
So thank you very much. First, I would like to thank the organizing committee for SIMPIC 2026 for inviting me today to join. Actually, this is my first time and for the SIMPIC conference. And the topic that I've been assigned to talk about is the research inspiration. So actually, I thought that, I don't know what I should talk about research management here, the ecosystem here, or you want the inspiration, use my career as an example. So what I'm gonna do actually, I'm gonna talk about my career as a physician scientist. And what are physician scientists? A lot of people here probably know. So, practicing physician who still spend a lot of time doing research, either health science research or biomedical research. So this is pictures of me doing clinic as well, which I'm still doing, almost every day, and working in the laboratory as well. Unfortunately, these species of doctor is vanishing, like you saw it on the article a few seconds ago. And what we do here is that we spend time doing both clinical care and research. And it could be, from clinical research to biomedical research. And typically, the physician scientist, in contrast to clinician educator, which are majority of our faculty members, we try to focus on disease-oriented, pretty much in basic science, and try to apply that back to the patients. And the place of practice is typically in an academic medical center like here or major university. We have to do clinical training and some additional research training. A lot of people do PhD, but for me, I did just a postdoctoral fellowship. I didn't do PhD, but I still, working in the lab and try to pursue career as a physician scientist. There's several challenges for physician scientists, and this is actually from the article from the US. As you can see, there are several challenges in the individual basis that in the US you have to apply for student loan to study medical school and also the PhD. So ended up that after you're done with MD, PhD, or PhD, MD, a lot of people have to pay debt. There's a constraint at the national level because of the cut of the funding from the NIH. And as you know, the current climate for funding in the US and everywhere in the world now is not really good. So challenges at that national level. At the institution, the number of people that interested in research, from students, from postdoctoral fellows, and also the junior faculty that want to do research is probably becoming less. So now back to my background. I graduated here from Faculty of Medicine, Siriraj Hospital, back in 1992. Fortunately, I graduated at the top of my class. My concept is to work hard and play hard. So when I'm in the room, lecture room, when I have to study, I just focus on study. But outside the classroom, if I want to play, I play hard. I served as the president of Siriraj Medical Student Union back in 1996 during my 5th year. I enjoy my clinical years, and I love learning from patients. I love to read more. I read review articles, textbook when I was a medical student, and that's not waiting for the faculty members, the instructor to tell me what to do. This is something that I love to do, and that is a good way to learn. I was interested in neurology during my 4th year, and my dream when I was growing up as medical students is that I want to have residency training in the US, finish the board exam, and become certified by the American Boards of Medical Specialties. And interestingly, even though I pursue my career as a physician scientist, I had zero exposure to research during my medical school years. Back 20, 30 years ago, research for medical students is something that, unheard of. Unlike today, that you have a lot of opportunities, and in fact, research is becoming, mandatory in the curriculum for our Siriraj medical students. Now, what are my inspirations? These are two of my clinical mentors that inspire me and want me to become physicians and neurologists. First is Professor Nippon Pongmarin. And as you know, Thailand is a small country, Southeast Asia. A lot of people don't know about us. When I went to the States for my residency training, a lot of people are still confused between Taiwan and Thailand. And we have to give credit to these professor. So he led a study that was published in New England Journal of Medicine back in 1987, using dexamethasone for intracerebral hemorrhage. And after he and his team show that it doesn't work. It cause harm to patients. So this is a practice-changing findings, and it's changed the practice since 1987 until now. Another mentor is Professor Narapon Prayundiwat, who actually shaped my early career until today. Another inspiration is Professor Pridama Lasit, and he's the one that inspired me to inspired me to become aspiring physician scientist. Again, he was among the first few people at our school that published in Nature Medicine, and this is the work in dengue hemorrhagic fever. And he set up his own lab here at Siriraj, and now his lab is still lasts until today and working on dengue hemorrhagic fever pathogenesis. So after I graduated in 1998, so I went to do a research fellowship at the Mayo Clinic in the United States. And this is my boss. So he's a professor of neurology and also immunology. So working in the disease called multiple sclerosis. And this is actually my first publication ever. So what we did is that we tried to study at the ultrastructural level using electron microscopy of the animal model of multiple sclerosis. It's a viral infection. And what we're trying to do is to quantify the demyelination, remyelination, and axonal damages here to see if it correlates with the neurologic functions of the animals at that time. And this is published back in 2000. Another paper that I worked with a group there is to prove that after repair, spontaneous repair by using antibody called remyelination, in these animal models, it can improve neurologic function. It's correlate nicely at an ultrastructural level by using electron microscopy. So during that time, between 1998 to 1999, so I spent time trying to study and finished the USMLE examination at that time and applied for residency training. And I ended up matched at Duke University, and with the help Professor Chirayu Uavarakun, who used to be a hematologist here, and then she went to become the dean of the Royal College, Chulapon Royal Academy Medical School. So I spent time at Duke University Medical Center from 2000 to 2007. And this is picture of Department of Medicine back then. As an intern, we have to wear white pants here. So very traditional program with a short white coat. And can you spot me? So I'm right here. Still with a lot of hairs back then in 2000. So that's when I was an intern. So, and then I finished residency at the same institution, became a chief resident in neurology, finished and actually tried to, achieve my dream, my childhood dream. And I finished the board exam back in 2005 and did 3 years of clinical and research fellowship in neuro-oncology. So this is actually a photo of me back in 2005. And there's several mentors, and this is important when you are, students, you are early career researchers, physicians, to have good mentors to help you, to guide your career. And this is, from several departments, from medicine, neurology, brain tumor program, and oncology. And without these people, I think I wouldn't have my career today as a neuro-oncologist. So I did 3 years of fellowship of neuro-oncology, and we call clinician investigator fellowship. So this is my small tiny office here. So what we did at that time is translational neuro-oncology research. So I do both clinical trials and also working in the lab and try to see if why these drugs work for these particular subgroup of patients. So this is the concept of translational cancer research, and what you can call bench to bedside and back. So it's kind of going back and forth between bench, a laboratory side, and also bedside, which is a clinic. So what we do here is actually first is the clinical trials. So we try to study the new drug that we have in patients. And this is several studies that I took part in using small molecule inhibitors targeting several signaling pathway in the most malignant, most common primary brain tumor in adults called glioblastoma, and including this one called bevacizumab. And you can see that the number of citations is really high, and the reason is that this drug is finally approved by the US FDA, and this is actually the first trial ever of bevacizumab in glioblastoma. And I was fortunate to be in that study, helping the design and also conducted clinical trials there. And also use a combination of these drugs, bevacizumab, which is anti-angiogenic agents, plus the targeted therapy against the signaling pathway called EGFR, the epidermal growth factor receptors, and this is in GBM as well. And like I said, one thing that I'm proud and I think is helping to see what we have done, actually reach the patient in the wider range, is that if you work on something and it receives regulatory approval, for example, this bevacizumab for recurrent glioblastoma, this is good for the global cancer community. And it's been approved since 2009 until now. So next thing is that we want to know why this drug works or doesn't work for a subset of patients. So the second thing that we want to go back to the lab is that we want to identify the biomarker of response or resistance. So this is a simple method that we use, the semi-quantitative immunohistochemistry, and try to see the angiogenic and hypoxic profile to see if it's correlated with response for patients that receive bevacizumab on the clinical trials that I just showed you. And what we found is we found some interesting thing that hypoxic markers predict response, predict survival of patients that receive bevacizumab. And now it's been proved in the large scale level using the genomic technology. So, and if it works or if it doesn't work, we want to try to test more. If we find the mechanism of resistance, we want to see if we test the concept of a new drug targeting the mechanism of resistance to see if it works. So what we're trying to do is to do the study in the preclinical stage using these multi-targeted kinase inhibitor, not only one single pathway but multiple pathway, and see if we can increase the efficacy in these patient population. Or using a stem cell as a model here. And we found one thing that is really critical is that bevacizumab, the drug that I show you, one of the mechanism of action is that it's probably disrupting the stem cell and stem cell niche in cancer. So, and before I left Duke, after I finished my fellowship, my boss asked me to write these seminal review article. Back then in 2007, it was state-of-the-art review of targeted therapies in malignant gliomas. And all of these failed to show benefit, with the exception of bevacizumab, that's still approved until today. So why study in the US for students who wants to pursue study, residency training, research training in the US? I think it provides excellent training in both clinical and laboratory research. We have exposure to technology advancement over there. There's abundant research opportunity and resources, both clinical, translation, basic research. We work with world authorities over there, which is important for career development and future when I return. So I still have collaboration, either in the education space, research space, or clinical service space as well. And we get recognized from the international community after we're done. So these are textbook chapters that I help co-author or author. And this is an example of opportunities that I received from my boss, from my mentors that I showed you before, to write chapters, textbook here, and it's been used globally. And after we're done, we still get recognized from the international community. I was invited as a visiting professor at UCSF and Harvard University to teach course and also to spend a week giving grand rounds and talking to residents, fellows, teaching students over there. And this is from last year, just to show you an example that you serve as an educational chair at the World Federation of Neuro-Oncology event. So we have 3 charter societies from Asia, from Europe, and also from North American. And this is an important educational event. So this probably not going to happen if I didn't, spend time doing a residency fellowship in the US. So now back to reality. So I returned to Siriraj in 2007 and start my career as a junior faculty. So I did everything similar to junior faculty here. So I spent most of the time, first 5 years, in the hospital with little time and actually little protected time to do research back then. Until 2013, when I know that if I, don't step out to have more time, to have more protected time to do research, I'm probably not gonna do research in my career. So, and neuro-oncology, I have to say that is a new subspecialty in Thailand and also for the whole Southeast Asia. There's only less than 4 dedicated medical neuro-oncologists for the whole Southeast Asian region, and a handful in Asia, actually. So this is a new emerging subspecialty. So, like I said, during my training, I trained to be a physician scientist. So in 2013, so I got opportunity from Deputy Dean for Research at that time and the department chair, allow me to, to choose the path of the research career. So I step out and try to set up my own lab. And several studies that I did at that time, clinical research, I participated in the vaccine research in glioblastoma. And these actually vaccine platform is actually developed at Duke when I was a fellow. And it's good that when I came back, these drug, this vaccine has been studied as a global trials, and we took part in these study and was published, even though it's a negative study, and but it's good that we offer hope for patients at that time. We did some studies that's sponsored by a pharmaceutical company, a new inhibitors at that time that was published. In terms of translational research, I tried to see if what we can do to improve the drug delivery to the brain, because this is one of the major challenges to treat brain tumor, because we have blood-brain barrier. These structures try to prevent toxin to get into the brain. In the same time, all the drugs that is being designed to treat cancer intentionally designed not to cross the blood-brain barrier to avoid the CNS toxicities. So what we're trying to do here is to improve the delivery. And actually, this I got opportunity from the Deputy Dean of Research at that time, and also Professor Siriraj here, who's used to be a director of the National Nanotechnology Center. And we had this project to work using nanoparticle and try to bring the antibody to get into the brain better. So at that time, we picked these disease called lymphoma. And what are clinical needs at that time, unmet clinical needs? We want non-invasive diagnostic technique, and also we want a new effective treatment strategies, particularly for patients that older or patients with immunocompromised state. So there's a drug that's been used for a long time called rituximab. It's CD20 antibody, FDA approved for several indications, autoimmune disease, lymphoma, but it has limitation in terms of blood-brain barrier penetration. So what we're trying to do is try to use nanoparticle to get into the brain better. And I worked with folks at National Nanotechnology Center, and I designed these particles and using liposome and also some coating materials, and try to bring these large molecules into the brain better. And we proved that in the in vitro and also the in vivo animal models that these antibodies can get into the brain better. And these received a patent recently, and it's been shown in the animal model of lymphoma that it worked better than the pure or unmodified CD20 antibody. Another project is the genomic oncology. And what we're trying to do, again, I have to thank you, Professor Siriraj here. When he was at Warsaw and National Research Council of Thailand, this project was funded by NRCT as a part of cancer genomic, cancer precision medicine. And we tried to do multi-dimensional genome and epigenome profiling. And also, we used a new technology called DNA methylation profiling. And these actually are now becoming more so a standard diagnostic technique for CNS tumors based on study by a group in Germany. And what they show here is that each cancer in the CNS has distinct signature of DNA methylation profiles. And what we did is that we use these grants to study, and we show similar findings to the group in Heidelberg, Germany. So they know us, and they invite us to be in a consortium and try to democratize this technology to be used worldwide. And we call these the Outreach Consortium of Molecular Neuropathology. And we're part of the steering committee and try to spread this technology to be used in real world globally. Coming back to our lab, we know that the methylation profiling to be used is very expensive. The ship is expensive consumables are expensive. So we try to develop an alternative testing to test genomic abnormalities and also the methylation using the third generation sequencing technology called nanopore sequencing. And this is, we're lucky to have Professor Tida Tip here, back from the US, and she's an expert in nanopore sequencing. And what we're trying to do and what we show here is that compared to the whole genome sequencing and the array that I show you from study in Germany, using nanopore-based copy number variation profiling, we can show a similar CNV profile to the gold standard whole genome sequencing here. And this is a one-day kind of lab, not like a 2 weeks of the traditional long read based methylation profiling. And we also developed the software because you have a lot of data coming up from this nanopore sequencing. So Tida Tip and team developed these software, and we use this data coming into these AI-based software and generate the report here automatically. And this is being protected by copyright, but we opened for academic center to use it. So now what we do here is that we have these tests up and running that detect methylation profiling using nanopore, copy number variation profile, and also the mutational landscape, the common mutations in CNS tumor. And we open these as a lab here at Siriraj, and we have samples coming from all over Thailand here. So this is part of the innovation we've tried to put here at Siriraj. So after 2017, 18, so I joined the research management team in the Center of Research Excellence here. And in 2022, I became a Vice Dean or Deputy Dean for Research and Innovation until today. So, but I still carry on some of the research projects, with the help of a lot of people in the laboratories. It's still ongoing, but maybe at a slower pace because of the administrative work burden that I have at this time. So with that, I think I'll show you the career from my medical school year, training, early research career, and now as a research leadership at Faculty of Medicine at Siriraj Hospital. So I think the career of physician scientist is important, particularly for countries like us here and also other countries in Southeast Asia. If we want to become a world-class science and innovation, we need people that know both clinic and also science. So physician scientist is an important career path, but it's not for everyone. For one class, I think if we have physician scientists, 5% is, we are successful. So if, you know that you want to be like this, physician scientists, get started early and try to engage during early years of medical school, try to focus research, find a mentor that, can serve as as your role models and mentors, and try to help you with small research projects during medical school year. And importantly is that time management skill is very critical because studying in medical school is tough enough, and if you add research project in your study time during medical school, it will become more challenging but not doable. a lot of students here I've seen can be successful in medical school, and also they do research in the lab as well, some of them. So it's doable, but it'll be challenging. And research, again, is a team effort. You have to learn to work in a team. So with that, I thank you for your attention.
Thank you, Professor Sid, for that thought-provoking session. The insights you shared today will, without a doubt, spark a deeper understanding of every participant here.
As a token of our appreciation, may we invite Khemjilat Hengsawat, the president of SIMPIC 2026, to kindly present a souvenir to Professor Sid. Now, moving on to our next session, it is our great honor to welcome to the stage Professor Emeritus Dr. Siriraj Songsivilai, Fellow of the Royal Society of Thailand and Chairman of National Commission on Science, Research and Innovation, who will be speaking on the topic Forever Young, why microbiology and immunology will be defining human longevity. Ladies and gentlemen, please join me in giving him a warm round of applause.
Good morning, everyone. It is always a homecoming for me to be here at Siriraj and to be here with you this morning. When I walk into this faculty and I see your face, young medical students, young faculty members, some old friends, it reminds me of my experience when I first came to this hospital in 1980. That was 46 years ago. So looking at you is also remind me of my younger self. What I'm thinking, what I was thinking when I were at your age, when I have the opportunity to sit here with you. When I was medical student, rather unusually, I'm not only focused in studying. I also got interest in research. Initially, not in experiments, but I keep asking unusual questions. And my mind just keep asking questions. Some of the questions might be with the students here, might sound even silly. And one of the questions I keep asking, was about life and death. It was about life and death. When I was second or third year medical student, in embryology, in physiology, we learned that human red blood cell, erythrocytes, can last, 120 days. This is what medical students learn, 120 days. And that every day, the body produces 200 to 300 billion new erythrocytes a day. It's 2.5 million new cells every single second. It also means that every second, 2.5 million cells die. When I look at the glass slide, I cannot tell which one are old, And where is the dead cell? How the body remove that dead red cells? Where is our garbage man? And I got that question burning in my head in the second or third year. So I decided to go into the lab. 46 years ago, I spent nights after nights in medical school. Actually, my lab was next door from this building, working on that subject. And fortunately, it took me about 2 years, until I get some of the answer of that question. And that question start the beginning of my career. Since then, I choose to work in a very new and the most advanced field of that time, immunology. So I became a faculty member in immunology. I have an opportunity to work on molecular immunology, on the development of human monoclonal antibodies and the antibody engineering technology back in Cambridge in the 90s. And it took 20 more years until therapeutic monoclonal antibodies are now transform medicine, change the way that we treat cancer. Immunology was a future 30 years ago. Molecular immunology was perhaps not even being heard of as the beginning of this subject. When I left Siriraj for the UK and for the US, I was in the Department of Microbiology, only to return the new Department of Immunology was founded. So things move very rapidly. But since then, I worked as the director of the National Nanotechnology Center, executive director of the National Research Council of Thailand, and also as permanent secretary for higher education, science, research, and innovation. Many colleagues, especially young faculty members, students like you, came to me for advice. The question is, which field should I choose? And I think this is very, very good question. Which field, as a young scientist, as a young student, should they choose to work on? And this is the most question that young people should ask, because that's going to stick with you for decades, and this is your life. My answer is simple. Pursue your curiosity. Pursue your curiosity. But more importantly, be in the right field at the right time. You have to be in the right field at the right time. Today, I will not talk about how to choose the field, but to cut all the story short, I will give you the answer. I will tell you the answer directly. Work in the field of the future. The field of the future, not the field of the present. You have to be in the field that will peak in about 20 years from now, when you perhaps will be at your best. Be in the field that is very new, very important. It is also very exciting. But what are they? What are these fields? One of the easy way to find out is to look what are the best minds, what are the best minds in medicine is now asking. What is a burning question that the best people in the field is asking. When I was here, when I was young, there were questions that my generation asked. But what is the question in your generation? But let me ask this. Let me repeat that question. How long can humans live? How long can humans live? Not how long we do live, but how long we can live. You have that number in your mind? 80 years? 100? 120? 150? Longer? I think that most people in this room would say 80 is so easy. We can live beyond 80. 100, we see many people that we know live longer than 100. The longest living humans on record was 122 years. So 150 is possible. It's actually very easy to do. And perhaps we will see the average life take longer than 150 years by the end of this century, and perhaps sooner. But 150 is, wow, this is something of the present and of the future. But let's look at history. Humans did not live that long before. Looking back in history, about 3,000, 4,000 years ago, and even up until the year 1800, the average life expectancy of human was about 25 to 30 years. 25 to 30 years. That's less than 200 years ago. It was increased to about 35 to 40 years around the year 1900. That is only about 120 years ago. Now, the overall global life expectancy is about 70 to 80 years. So in about 4,000 years, we are able to increase life expectancy from 20 to 40. We add only 15, 20 years within 4,000 years. But in the last 120 years, we almost double the life of human in a very, very short time. That is a miracle of modern medicine. But why does happen? What killed most human in history? Because we are in the student class, I have give you a multiple choice. What is the major cause of death of human in history? Aging, cancer, heart diseases, strokes, war, infection, or malnutrition. What do you think? What is the result why we die so young over the past 4,000 years? The answer is clear. Infection. Infection killed most humans in history. And the major cause, tuberculosis, diarrhea, smallpox, malaria, and sepsis. Malaria, that plasmodium perhaps killed about half of human being born. So it have killed billions of humans. Before modern medicine as we know it, most humans never live long enough to experience aging. So not because we are weak, but because humans die too early from infection. And that's happened in the first 20, 30 years of life. And what changed that? It's not a single invention. It's not a single drug. It's a series of things. So modern medicine didn't make us live longer. Actually, we have potential to live longer than that. Modern medicine prevents us from dying too early. And it's not a single invention. There were major breakthroughs over the past 200 years. Sanitation, clean water. This has happened around 1600s onward. We know how to make water clean. We know how to separate waste from the food that we eat. The air is cleaner, and so on. Vaccines, antibiotics, and improved nutrition. The development of vaccine by Jenner in 1796, starting with smallpox, eradicate smallpox, and subsequent vaccines do the rest. Antibiotics, Fleming in 1926, 1928, starting with penicillin, and then better and better antibiotics coming out. We know how to get good food, how to get enough protein, enough energy. And these in combination add about 40 years of life to everyone on earth. This together. And as you know, this is our achievement as microbiologists, as immunologists. We know how to kill bacteria. We know how to prevent virus. We know how to kill almost all parasites. We know how to prevent disease from vaccines. All the magnificent tools have been developed. Our job is done. Our job is done. Are there any other jobs for microbiologists, for parasitologists, or even for immunologists? We are now left with some rare disease, extremely rare disease. We, from time to time, work on emerging and re-emerging infectious disease. So, as the future immunologists, future microbiologists, future doctors here today, what would you do? Those success, our ability to add years of life also come with a cost. It's not free. We have eliminated not only harmful microbes, we also kill almost all microbes with antibiotics. So the good ones also gone. And on the other hand, there are emergence of antimicrobial resistance. We're now facing multi-drug resistance from time to time. And more importantly, we have disrupted the balance between ourselves and our friends, the microbes. And with that, we also die. We do not die early, but we die later. We do not solve the problem of death. We solve the problem of early death. And someone said that we change the disease that we die from. The issue is that the current medicine issue is that we now face aging. Because humans do not die early, we die later. Modern medicine do only prevent early death, but we have not yet slowed the aging process. So when we live longer, there are new diseases emerge. You know, cardiovascular disease, hypertension, strokes, and so on, diabetes, cancer. And if we live even longer, we will get neurodegenerative diseases. And if we live even longer, there might be more diseases we still not yet known. This is now called NCDs, non-communicating disease. It is nothing to do with microbiology, nothing to do with immunology. As lifespan and health span have been increased significantly, there are also some worries. We see that in the last 120 years, we add 40 years of life. Can we add another 40 years in 100 years? Or can we go beyond that? It will be life forever. The question is also now that will human can live beyond 200 years? And there are many thinkers said that if we live beyond 200 years, we might live forever. And can we become forever young? Can we be young forever? So modern medicine is shifting. It's shifting from infectious diseases. During my time, infectious disease is still important. But now, the modern medicine has shifted. It's shifted to how we deal with aging. Can we delay aging? Can we repair the aging? Or even can we reverse the aging? So the current directions of medicine, especially in longevity, is now working on the field like biomedical engineering, starting from, for example, prosthesis, replacements, and so on, in AI, in robotics, in how humans can work with robots, how the biology and mechanics can work together. Very exciting. On cells and tissue engineering, on CAR T cells, on new and better monoclonals, on genomics, precision, personalized medicine, and on preventive medicine. These are the fields that you've been heard of quite often these days. But let me tell you some insights, some very secret, especially for future doctors, for future microbiologists, for immunologists. The idea and the main concept is about to change. The future of medicine is not about killing microbes. It's not killing microbes. We have killed microbes in the past 200 years. It will be about learning how to live with them. And the concept of living and utilizing microbes to extend life is starting. We now start to understand, and there are increasing evidence that chronic inflammation is central to longevity and many diseases. And if you look at new and new articles, there will be comments like chronic inflammations disrupt the systems, create regenerations, and so on. And as a result of small, continuous inflammation, we age. So by working, controlling, moderating chronic inflammation, this is central to longevity. And as well, the interplays, the interaction between infection, inflammation, and the immune systems all together will markedly define the life, the limit of our health span. Working all together, everyone wants to be young forever. But forever young may not come from anti-aging drugs alone. We might not have a single pill that extends our life. Good exercise maybe, good sleep maybe, good nutrition maybe. But it is very likely that it will come from our ability to control infection, especially to control chronic infection. Our ability to understand and to work with microbiome. Our ability to regulate immunity. And taking all of that together, our ability to engineer, to regain a friendship with microbes. Why? Because within our human body, there are more microbes within us than the total number of our cells combined. So, in the past, longevity is equal to avoiding early death. Microbiologists, we kill pathogens. We find and kill pathogens. We identify that this is Staphylococcus, and then we have the right antibiotics to treat them. Immunology, develop vaccine, fight the enemy. But in the future of medicine will be on modifying the aging process. For microbiology, how we coexist with the microbial ecosystem. For immunology, how we could regulate and control inflammation. And how we rejuvenate our immune functions, taking out the waste, metabolic waste, old cells, bad cells, and also keeping our immune system young. Everyone here, you are the one who can shift this paradigm. This is very, very new paradigm. Do not think microbes as the enemy. We are an ecosystem. We are the ecosystem. We have ourselves, we have our friends, we have our enemy. We have bacteria in our body, we have virus. So it's changed from the war model. The war model is that you are the enemy. We have to fight you, we have to kill you. The ecosystem model is that we live together. You're doing something good for me, I'm doing something good for you. And in doing that, there are a number of changes that we have to make. And central to that, immunology would be a master switch that look at and control all of the interaction. So let me tell you what I'm thinking about new microbiology, new immunology. It will be about balance. It will be about balance. It will be about precision balance. How we could work and motivate the whole system happily. We have to see human as microbiome ecosystem. We cannot separate. This is bacteria, this is us, you are our enemy. We cannot live together. But there are increasing evidence that there are many microbes within the bodies. There is a recent article that some microbe synthesize something that help delivering good metabolites into the brain, for example. And also, we have to look at immunology as the core component. This is a new term, inflammation, and aging all together. This will change the way medicine is practiced. The use of microbiome across organs. There are many bacteria in the gut. There are lots of that. And because of the use of antibiotics nowadays, many of the bacteria, good one and bad one, are killed. We have to restore what's in there. We have to be able to selectively restore that. We have to develop a system to delay or reverse immunosenescence. We have to be able to control inflammation. So the future of microbiology and immunology will be about precision. Will be about holistic approach. Will be about targeting, precision plus balance. And this is a new frontier. If you start in immunology or in microbiology today, I would recommend that this will be your new frontiers. The ability to work on microbiome engineering, how we would be able to understand each and every microbes in our body, how they work together, how they work with us. What are they good for us? And what we could be good for them. The other new frontier is on immune engineering, but not on human monoclonal antibodies that I work on, but on ability to moderate, finding a system that can identify, to remove, to put away bad metabolites, keep information in a good way that protect us and also not causing any more damage. Rejuvenating the immune systems. Keep them very young. Keep them work together. And these are the very hot field that I guess it will peak in about 20 years. Our ability to control immune response will provide our ability to control aging. So that's perhaps return to my initial questions about life and death, about when I look at the slides of the human red cells. Why will microbiology and immunology will define human longevity? Why? Because the future of human health span will be determined by one thing. Our understanding and our mastering of the relationship between microbes and our immune system. If we know that secret, and if we control that, if we can work with that, it's very likely that we will be forever young. So for students, in a few years when you become doctors, for faculty members when you work on that, future doctors will not only treat disease. You will not only treat disease. You will be an engineer. You will be the designer. You will be the engineer of the microbiome ecosystems. You will be the designer of the immune interaction and working together. We will work in the new world system that is the human microbes hybrid relationship. And with that, I think I have the opportunity to talk to you that the future of medicine is changing. And the future is yours. Thank you.
Our deepest gratitude to Professor Dr. Siriraj for his profound and insightful presentation. It is truly our honor and privilege to have you here with us today, and we sincerely appreciate your generosity in sharing your invaluable expertise with all of our participants.
We will now proceed to a Q&A session and take questions from the audience. Please feel free to raise your hand. We warmly encourage anyone with questions to take this opportunity to learn. A mic here, please.
Yes. Okay, please.
Thank you, Professor Sheet and Professor Suraj, for your amazing description of your career history. Really were very inspired from your speech. So we are from Bangladesh, actually. We joined along with my two colleagues, Professor Arif and Professor Abdullah Akhtar. So thank you so much, especially for the neuro-oncologist. You know that basically the clinicians are not so much interested in research. So, but you made in such an amazing career for you, and then your speech inspired us to join this one. For developing countries like us in Bangladesh, actually, how we can send the clinicians to the labs, because usually even the clinicians in our part, they cannot hold the pipette itself. So this is, I feel, some lacking in the physicians to going to the labs. So is there any special medication for them to be inspired in the laboratories, in the labs to work?
So, like I said, well, during my time as a medical student, I had no exposure at all back 20 more years ago. At this current generation, at medical school has some mandatory courses for research. So we do 3 years during the second year, third year, and fourth year. And they don't have to complete the research project, but at least they know how to design the project. They know, basic nuts and bolts about doing research. But some of them will, do like systematic review meta-analysis, which is, a good way to do research as well. But I think, coming back to your questions, how do we encourage students to work on?
So this is in the
lab, right?
undergrad curriculum included.
Right. This is our 6-year MD program. So we have, it's in the curriculum that all medical students have to do research. It's not elective, but it's mandatory courses. Yeah. So I think this, some of them will choose mentors that work in the lab. So at that time, they will, have a chance to work on pipette, doing, Western analysis, Western blot analysis, doing genomic testing and all this stuff. But to be honest with you, if they want to do that, typically they have, they know that they want to do research. And a lot of our students, actually, when they're in high school, they're in the school that had a research project already. They know how to use pipette and everything before. But yeah, for me, I didn't know how to do it until I finished 6 years. And my mentor sent me to the lab and tried to work on it before I was shipped to the United States to do research. So I think try to encourage them to see how important research and innovation is, and why we need to do it. It's important for future diagnosis, treatment, and everything. If we don't do it, we're not going to move, the field forward, particularly for diseases that is seen locally, a niche in your country,
Thank you so much.
Thank you.
And another question for the immunologist. Sir, you have shown us some visionary aspect of the thinkings, actually. For example, how should be the future prospect in microbiology and immunology? So for example, we should be visionary, and we have to some new dreams, actually. Without dream, we cannot reach the target, and we cannot make a new consensus, actually. So like as an immunologist, if you are referring to some process regarding the aging process, how can be reduced, or is there any research going on regarding this one, immunologically or microbiologically, how the aging, the challenges can be fitted? Thank you.
I think that the issue is choosing the right field to work on. That is the most important decisions one have to make, especially in the early career. So doing something that would be at the peak some years from now, especially when I talk about immunology relating to aging, there are increasing evidence that perhaps 2 or 3 main evidence that I'd like to talk to, that the inflammation, chronic inflammations actually worn out our body systems. So, and that in itself, plus some metabolites and that kind of thing, is the main core of the aging process. And also the repeating cycles of being killed, born again, and that kind of things is a result of some diseases like cancers and with plaque and a number of things. So there are very solid scientific evidence that back up on the relationship between the aging process and immunology, and also with the chronic infections by some bacteria and virus. you.
Okay, thank you so much.
Welcome.
My last question is to the onconeurologist. So what antigenic preparation you are using for gliomas?
I'm sorry, can you repeat it again?
What antigenic preparation you are making for making the vaccines against the glioma?
Oh, so that one,
yes.
that's EGFR
You had one
V3.
paper article regarding this one.
Yeah, so it's the EGFR V3 peptide.
Peptide.
The peptide that mimicking the most common mutation of EGFR called, EGFR variant 3. Yeah.
Okay, thank you so much.
Okay, so previously about what interests medical student in research, as a second-year medical student here myself, I think what interests me is when my professors come up and talk about how they started with research questions and then how they actually tested those questions and apply it to real patients. I think that really do interest me in research and intrigue me into spending more attention in research classes. So I think the application or the stories about research really helped me. And I have a question about immunology. You say that in the future we should design new interventions to contribute to anti-inflammaging. I want to know how to start designing the research, because right now it seems a bit far-fetched for me.
Okay. Think that first you have to find something that would interest you. Each one of us have different interests. Because we ask some questions, other people are not interested in that questions, but some other people's questions may not be our interest. So in order to start your career, you have to work on something that you are really interested, because it's going to be with you for years. And as researchers, especially as the people doing experiment, there would be more failure than success, days in and days out. So you have to be able to withstand that failure. And so if you are not so interested in that subject, many people will give up because you will not get result in time that you are happy with. That is one thing. But once you find something to work on, the best thing is to find people, to find the faculty members, to find professors who would guide you through that process. It is mentoring that is very, very important. So be in the right place, choose the right field, and find the right person that you want to work with. And then once you find that, the rest would be more or less easier. So my advice is that keep asking yourself what you want to work on, and then look around, find the people who work on that subject, who work. If it's so new, there might not be many people who work on that. So, but you'll be able to find, and then talk to him, or talk to her, and then follow that path. This is what I did. This is what Professor Sid did in the past.
I'm Xi Wai from National Yangming Jiao Tong University, Taiwan, and thank you for your wonderful talk. And I have one question: is that since many studies talk about microbiota composition associated with the longevity. For example, some bacterial species may contribute to the extension of a lifespan, such as Akkermansia or other bacterial species. But according to your talk, you also mentioned about microbiome engineering. But in Taiwan, we also try to use a single species to test whether they have ability to extend the lifespan of the mice, and also we for reality, actually for the FMT can be only used for the treatment of some specific disease like Clostridium difficile infection, and the composition is very complicated. So in your opinion, which will be the best strategy to do the microbiome engineering and then to contribute to the longevity? Thank you.
Thank you. That's a good question. The medicine today have changed. It's changing from a single component, single drugs approach. When we look at, for example, in genomics, the single genes related diseases or condition is good, but this also we now know that in order to have a certain phenotypes, it's a combination of interactions of so many genes. I think that's something similar to microbes. So currently we have the ability to work on microbiome, not only on a single organisms, and it is very likely that the combination and ecosystem of microbiome would have an effect than a single component. And that is also a core of what we call oriental medicine. It's a combination of so many things, not a single component. There are several ways to work with that. For example, there is an ongoing work on some people who have super long life. It's called super agers, people who can live happily in their 100, 110s in a good health. What they have in their bodies? What are the bacteria or viruses that they have in their body? We now have tools to look at, for example, gut microbiomes. We know exactly what are species of bacteria in the guts of that people, and so on. I think that taking in combination, it will increase our understandings on what's happening in our body. It is in a very early stage yet. Thank you.
Yeah, thank you.
We will accept one more question before moving on.
Hi, so this is a question for, I think, high schoolers. So I've seen how both speakers really branched out through their entire careers, going to different universities and doing research. And I've seen especially the citations where I've seen so many authors on there. And I think just as a high schooler, I'm really wondering how you guys managed to, I guess, social network and find peers with common interest and research on that specific topic.
It's increasingly more and more the trends that now, in order to do a single work, this a combination of works of many people. So you will see many authors in a single scientific paper. This is now common, which shows that many of the work would need so many people to work together. In order to start that, normally we start small. So the network do not build in a single day. You work, as I said, especially when you are young, you should not start working alone. You have to work with someone who already know that subject. And that someone would know someone who work in that subject. And that someone will know some other people who can add dimensions to that. And that is how the network starts. There is no need to rush. It will come naturally when your career develops.
Thank you so much.
Thank you everyone for those thoughtful questions, and once again, thank you Professor Dr. Siriraj for those insights and for being with us today. We would now like to invite Khemjilat Hengsawat once again to present a small token of appreciation.
And now, from the foundations of research and essential concepts of microbiology and immunology, we now move forward to explore its impact on clinical science. We are delighted to invite our third speaker, Associate Professor Dr. Patcharapan Teshasinthana Sarasombat from the Parasitology Department, who will share her expertise on bridging clinical needs with research, lessons learned from neglected parasitic diseases.
Okay, so good morning, everyone, and thanks again for inviting me to give such a wonderful talk today. So today I'm going to talk about my research and also my research journey, entitled Bridging Clinical Needs with Research, Lesson Learned from Neglected Parasitic Diseases. So before I begin, I would start with this slide. Uh-oh. Okay, sorry. Oh. Okay, as a physician, this is the thing we counter. It all starts here. You know that there's many times when we work as a physicians, we have to break bad news to all the patients and family. So the things that you do is that you're going to them and then say that, I'm so sorry, there's nothing we can do. And that's all we can do. And keep saying sorry. At that time, I feel like I'm a Napoleon Bonaparte sitting on an Iceland and then thinking about myself, whether there's nothing we can do for sure or not. Many times you have seen patients and you follow all the guidelines and there's nothing we can do. So I started asking myself, is it real that there's nothing we can do for the patients? And that's my story begins. So today I'm going to talk about my research journey and also what we have learned throughout my career, 10 years. I'm going to begin with the first episode, which is the episode of the pneumonia story. Having that questions in my mind, I think that there's more things we can do besides saying that there's nothing we can do. I decided to pursue my PhD because I think that research and innovations will actually help us improve the healthcare and answering all the questions, whether is it real that there's nothing we can do. When I got back from my PhD, the thing that I did is that I did not even have any lab equipment or nothing. We have only the pipettes. So, as a physician, I feel like, okay, is this the things that I really want to do? I started my career with the easiest one, which is to do molecular diagnosis. Actually, I got my PhD in immunology, so switching gear to do some kind of molecular diagnosis is kind of challenging, but I learned things along the way. So started with pneumonia story. It's about the disease called pneumocystis jirovecii, pneumonia. I think you guys may familiar with this. Pneumocystis jirovecii is a fungus that causes opportunistic pneumonia in the patients who have weakened immune system, like patients who have HIV or like patients who are on immunosuppressant drug or have multiple problems that like transplants. You get people here. Actually, this disease, you have been exposed to this organism since you probably were like 2 years old by the age of 2, but you don't develop any diseases because your immune system is good enough. However, like if you have weakened immune system or you're neonate, you tend to develop pneumocystis pneumonia or PCP. So You might be familiar with PCP in HIV. When I was in training like 20 years ago, HIV were so abundant, and at that time, antiretroviral drugs or prophylactic drug was not available. So the majority of the people who have PCP are HIV. But nowadays, things change. We have better care for the HIV living, the people living with HIV. So other immunocompromised patients are the group that develop PCP. Is the majority of the group that develop PCP. But unlike PCP in HIV, people who are non-HIV develop the disease more severe, abrupt, and acute. The problem is that at that time, the diagnostic test that available is the conventional lab that I will mention later, and the clinical pictures of the patients who have PCP in non-HIV are really atypical. So in contrast to HIV patients, will have just a typical pattern of chest X-ray and also like CT scan that we might observe reticulonodular infiltrations. Patients with non-HIV, when they develop PCP, the clinical manifestations most of the time is similar to other diseases, other pneumonia from different causes. And the problem is that the laboratory investigations that we have right now for the conventional laboratory method have really low sensitivity and specificities. And also, there's a problem with the specimen collections. Patients who have PCP, the best type of specimen that we use for diagnosis of PCP is bronchoalveolar lavage fluid. But as you know that in non-HIV patients, there are some people that cannot undergo bronchoalveolar lavage. For example, people who have like hematologic malignancies that probably are not fit enough to perform bronchoalveolar lavage. So there are two main problems. The first one is that the conventional lab that we have right now is not really good enough. And the second thing is that the specimen type that used for diagnosis is invasive specimen. And another challenging thing is that if we develop the test that has really high sensitivities, we'll pick up the patients who actually have the colonizations of this fungus in their lung without having any diseases. You can see that like the fungal burden is correlated with the clinical manifestations. So people might have only colonizations or subclinical infections of this disease. But if we decide a test that's really sensitive, those patients will be diagnosed with PCP. However, the conventional lab that we have right now can only pick up the patients that have really high fungal burdens. So there's a problem with, there's a gap in here that there's some patients that actually they have active PCP but cannot be diagnosed with conventional technique. That's the reasons why we come up with the molecular diagnostic technique that can actually tell the fungal burden of the fungal load of this Pneumocystis cerevisiae in the specimen, and we're trying to define the cutoff point to distinguish patients who have real true PCP from colonization. Okay, so you may ask whether there is any commercial test available for this so that we don't have to develop our own in-house quantitative PCR for diagnosis of PCP. Of course, there are some though, but they also have the same problems. There's some commercially available, but the things, it's like the COVID test kit that we know, right? So they would interpret results as only positive or negative. They cannot tell the difference between the amount of the fungal burden in the specimens. That's the reasons why I came up with the first work that we did, trying to find that cutoff to distinguish the patients who have PCP in HIV and in non-HIV patients. However, there's still problems though, because the first set of the experiment is the retrospective experiment, so there's some limitations about like how to the clinical correlations with the amount of the fungal burden we find in the specimen. And another thing is that we don't know whether we can use non-invasive specimen like sputums for diagnosis of PCP or not. That's the reasons why we started doing the next kind of study to determine the optimum cutoff values for to distinguish active PCP from non-PCP in both invasive, which is bronchoalveolar lavage fluid specimen, and non-invasive, either from like tracheal sputums or induced sputums. And the second thing is to evaluate whether the fungal burdens in non-invasive specimen and invasive specimen are the relationship between these two, whether when we have high fungal burden in the lower airway, whether we see the same thing in the upper airway specimen or not. That's the second work that we did, which is the prospective study to compare the fungal load in paired sputum samples and bronchoalveolar lavage fluid for diagnosis of PCP. In this study, we recruited all the immunocompromised patients presented with pneumonia, and we collected the samples from bronchoalveolar lavage fluid, which is the invasive specimen, and also collect the non-invasive specimen from the same patients within 24 hours apart. So non-invasive specimen can be induced sputums or tracheal sputums in case that patients were on endotracheal tube. And after that, we did some conventional study conventional technique to detect pneumocystis cerevisiae in this type of specimen, and also measure the fungal burden in both type of specimen. And we work with the clinicians. So the clinicians will actually categorize patients into 3 groups: definite PCP, probable PCP, and non-PCP, based on their conventional, their clinicals and also conventional lab though. And we further evaluate the amount of the fungal burden in both types of specimen, as well as to study the correlations between the fungal burden in both types of specimen from the same patients. And this is how we classify the patients into definite PCP, probable PCP, and non-PCP. The difference is that in the definite PCP, the patients have to have at least one of the conventional tests that is positive. For probable PCP, the patients would have the clinicals that similar to PCP, except the conventional lab were all negative though. And after receiving the treatment, they improve. And for non-PCP, the patients will, the clinicians think that they are less likely to have PCP. And the patients is divided into, we recruited 122 patients and 8 were excluded. These patients will divide from, based on their clinical criteria, from PCP and non-PCP. And in PCPs, there's 11 patients that met the definitions of definite PCP, and 12 patients fit the probable PCP criteria. And here we came up with a cutoff, which is the cutoff to help distinguish the patients who have PCP from non-active PCP from colonizations. And that's the sensitivities and specificities of the test. the ROC curve analysis also show that it's really reliable because area under curve is really close to 1. The next thing is that we asked whether we can get this cut off from non-invasive specimen or not. We found that in the non-invasive specimen, we get a lower cutoff number, but it still uses moderate sensitivities and high specificities for diagnosis of PCP. We further observe the correlations, whether there's a correlation between the specimen from lower respiratory tract and upper respiratory tract or not. We did both correlation study, Kappa coefficients and Spearman rank correlations. And we found that for Kappa coefficients, though, there's a strong correlation between the fungal burden in tracheal specimen and bronchoalveolar lavage fluid. And for Spearman rank correlations, so the overall correlation is good too. So another systematic bias check is we did perform Brown-Adman plot with, to see whether there's a systematic bias between the amount of the fungal burden that we found from the upper and lower airways or not. And the things that is very interesting is that in 4 patients, we'll have 2 patients will have definite PCP and 2 that have probable PCP. They actually have a really high fungal burden in bronchoalveolar lavage fluid, but really low level in induced and tracheal sputums. That means that this can be interpreted that the invasive sample offer like high sensitivity and specificity for PCP diagnosis. While non-invasive sample show high specificities, but the sensitivity is kind of moderate. So to interpret that, it means that non-invasive samples probably is good for to help confirming the diagnosis, but not for screening. From here, it can apply for the clinicians that if the patient is high suspicions of having PCP, but the fungal burden in non-invasive sample is below the cutoff point, bronchoalveolar lavage should be performed. In case that the clinical is highly suspicious of PCP, but the non-invasive samples fungal burden is above the cutoff, you can diagnose with that patients with PCP. And in case that if the level in the non-invasive samples is below the cutoff and the patient is clinically less likely to have PCP, so PCP is less likely. And if the patients will have clinical is not compatible with PCP and non-invasive sample is above the cutoff, that patients may actually have PCP, so they may be useful from receiving anti-PCP drug. Okay, and my dream is that I also, we actually have this in-house test available to use in Siriraj Hospital right now. I started from July 1st, and as you can see right here, last year in 2024, we have only 90 samples, but in 2025, it's increased to 376. So there's a significantly increase in the test that is available. And the things that I really happy about this is when the physicians call and tell me that, wow, it's good that this test is available for use at Siriraj Hospital, because sometimes there's a patients that the clinical pictures cannot distinguish whether that patient has PCP or not. And having this test will help them diagnose, confirm the diagnosis as PCP or non-PCP. And there's one patient that can discontinue the medication, so they don't end up having a side effect from receiving anti-PCP drug. And people may ask what is next about this project though. Yesterday, I prepared a slide, and then my two boys came to me and asked me, like, Mom, what did you do? So I asked them to help a bit with this slide, and why there's so many things on this slide. So the next step is that since the first prelim data look very good though, so we're developing a test with a different technique. It's multiplex qPCR, and then we are patenting this. And yesterday I did not come up with the name of this test yet, so I asked my son, okay, how should I name it? And they said that Mom should name it Hi Nemo. So that's why I would call the multiplex qPCR that we are patenting like Hi Nemo. And hopefully not only the patients at Siriraj Hospital will be able to use this test. And then whenever you go to clinic and you think of PCP, you have to think of me. And patients in a different part of the world will be able to purchase this test with a new one to, so that we can help with diagnosis of PCP, not only at Siriraj Hospital though. Along the way, I received an email from a company, a medical diagnostic company, asking about the technique, but I know that they're trying to develop their own. They're asking about some of the cut-off value and how I calculated it. So it means that there might be some like potential marketing availabilities of this test. Another things that we are doing right now, so we're trying to see whether we can develop the tests that have the specific state-specific diagnosis, because we know that the state of the disease, the parasitic state, even the cystic form or trophic form, is correlated with the clinical outcome and disease severities. And that's why that's another things we're developing right now. It's called Immunology. It's the kit to detect whether the patients will become severe or less severe. So my son named it. That's the reason why they said that Mom should name it New Monitor. So the minute I heard it, I think of monitor lizard also, and then they helped me put this monitor lizards and Hi Nemo here on the slide. Okay, so that's the first things I wrap up with my work on the first episode though. After working as a scientist, a researcher for a while though, I actually become really interested in the free-living amoeba. I would name this second episode, the free-living amoeba world. So if you have heard of amoebic brain-eating amoeba, that's one of the amoeba in this group though. So when I work at Siriraj Hospital, we have observed the patients having this disease coming, like this one is the 4-year-old woman developing meningoencephalitis, and this is from her CSF, cerebrospinal fluid. And we know that this disease is really fatal, and that's the first case. Normally, when you read like a textbook, it would say that this disease is very rare though. But after a certain period of time, there's another case, the 4-year-old boy developing primary amoebic meningoencephalitis, and this is from his CSF. And if you heard of the primary amoebic meningoencephalitis, there's one Korean guy that used to work in Thailand, and when they went back to Korea, they developed meningoencephalitis, and that's the first case in Korea that received exposed to this amoeba while he was in Thailand. At that time, the Ministry of Public Health called me and see whether I can help identify the source of his infections or not. And we help with that and then identify the source of his infection so that we can prevent other people from having this fatal disease. But the story hasn't ended yet though. There's another case 4, case 5, from Khon University and more. So at that time, though, there's a woman in Texas and in Missouri having the primary amoebic meningoencephalitis too. And the answer to all of this is that they all pass away. That's very sad though. That's why I become interested in studying this disease, because I want to answer the questions, is it real that there's nothing we can do? Talking about free-living amoeba, there's 4 genus that cause the disease in human. And this genus, Sapinia pedata, there's only one case worldwide, so that's not in the study focus. But these 3 free-living amoeba, all of them is really fatal. Whenever this amoeba infect brains, you can only pray though 95% of the patients die, but 5% of the patients survive, and that's my research questions. What make them survive? And why can we actually make these treatment options better, and besides the diagnosis? So for Neisseria fowleri, though, I would, actually there's a cases in Thailand and in many parts of the world, though most of them are not reported. That's the reasons why the incidence might be lower than what we expected. Starting with the first amoeba that I show you, is Neisseria fowleri. So people get infected during water activities or when they use tap water for nasal irrigations. This amoeba like to travel to their nose, right, and then cause severe brain inflammation. And that's all the medications that patients receive right now, and all of them, most of the drug treatment is limited. And even if we give them multiple drug treatments, the patients still not survive. And there's other interventions that we're trying to answer whether it's benefit giving to the primary amoebic meningoencephalitis patients or not. That's the gap and opportunities to improve so that we can answer whether there's nothing we can do for sure or not. And this is the first draft of my graphic abstract, and hopefully this will come out soon. So we did the experiment with high throughput compound sequencing from commercial available drug, though, and then find the lead candidates in vitro, and then do cross-strain validations with another clinical strain that we obtained from the patients who have primary amoebic meningoencephalitis. If you're a doctor and then you end up with diagnosis of primary amoebic meningoencephalitis and you let it go, there's no expanding knowledge in this disease. But the good things is that we can actually isolate those strains to study for drug responder. I did this response setting up the criteria that we used and came up with 2 lead candidates that we did the mouse model to test the efficacy of this drug. And that's the amoeba that we recover from the mouse model that we have. And to answer another question is whether therapeutic hypothermia can help with treatment of primary amoebic meningoencephalitis or not. So we invented the therapeutic hypothermia. It's like an Arctic sun that you will see when you in the ICU for human though, but this is in the mouse version. Besides that though, there's a free-living amoeba. The second one is Ballumutia mandilaris. I received a call from King Mongkut Hospital like 6 years ago almost though. That patient, she was suspected of having amoebic brain infections, but it's a different form though. So she has the thing called granulomatous amoebic encephalitis. We had a chance to actually help diagnose this patient, and we successfully established a new strain of Balamutia mandilaris from that patients by co-culturing it in a lung mammalian cells culture. And we transfer it to a BM3 media, which is the arsenic media for studying of the drug though. At that time, since this disease is chronic, I have a chance to meet this girl. She's a lively girl though, and her nickname is Kaimuk, a lovely pearl in Thai. So that's the reason why I named this strain KM20. And KM is also King Mongkut Krao Hospital. That's why we established the strain. We talked to her mom, and her mom said that she doesn't want anyone to have this disease again, and that left a mark in my heart though. I will do all the best to try to find a way to cure patients with free-living amoeba. So that's come up with the knowledge obtained from this strain that we collaborate with Chinese University of Hong Kong. We did high throughput screening. We entering the mouse model that we just established in a in immunocompetent mouse model. And I still have other projects going on. It's about circular RNA vaccine for parasitic diseases, and I also help the molecular diagnosis of the neglected parasitic diseases. I have like 10 minutes left though. I would like to acknowledge all my lab members, especially Dr. Nathawat Chayawong. He's like my brother to me though. And then like we did all the experiment together. Then my team, I would like to acknowledge the patients who involved with the PCP projects, these two young clinicians, Professor Park Poom, Associated Professor Dr. Park Poom, and Associated Professor Dr. Nduk Pop, for helping me with the clinical study. We also obtained the funding from Siriraj Hospital, my Faculty of Medicine, and also from the Medical Council of Thailand. For the free-living amoeba projects, though, we collaborate with Dr. Niwat from Faculty of Science, Mahidol University, Dr. Monlat Julanate, Dr. Lapon Anopat from Department of Parasitology, Faculty of Medicine, Siriraj Hospital, Professor Stephen from Chinese University of Hong Kong, Dr. Natapon Taninton from Washington University School of Medicine, USA, we received the funding from National Research Council of Thailand and also Anantam Hidon Foundation. From the circular RNA that I did not have time to talk about today, we collaborate with a big team, Professor Suradet Hong Eng from Faculty of Medicine, Rama Thibadi Hospital, Professor Pathompon Wongkongket from Faculty of Science, and also Assistant Professor Yanin, Associated Professor Poom from Faculty of Tropical Medicine, Dr. Niwat, Dr. Waralat from Faculties of Medicine, Rama Thibadi Hospital, and my team here at Siriraj Hospital. And also, right now I have another positions, which is the head of the Department of Parasitology, and that I would like to thank my adventure team that make things possible. And last but not least, though, I would like to acknowledge my patients and their family that inspire me to do research. Laboratory animals that have been used present day or in the past, and also my family that's newborn. So our mission is that neither persons nor disease should be neglected. That's the reasons why we form a small team called SICNPD, Siriraj Integrative Center for Neglected Parasitic Diseases, because we don't want the things to end at bedside. And last but not least, though, is an ad town. So for the people who are interested in research, innovation, and also translations to clinic, though we have this program, MSc and PhD program in biodesign in medicine with the collaborations of Faculty of Medicine, Siriraj Hospital, Faculty of Engineering, Mahidol University, and Faculty of Medical Technologies, Mahidol University. For this stage, we use the biodesign innovations process for need identifications, invention, and implementations in order to translate the thing from bedside to commercializations. We have 3 tracks: biological therapies, medical device, and digital health. And you all are welcome to check out the website and then to join us as well, though. We help with the discovery of the idea, development to deployment. And I was once asked whether, and challenged whether I will be able to kill the patients with free-living amoeba or not. My answer is that I don't really know, but I will try my best. The things that when I look back, though, I'm not the first one who trying to kill this disease from free-living amoeba. There's Professor Visweswala, Professor Structure, who actually work at CDC that inspire me, though. They inspire us from whatever I have read through their paper. This is just to pave the way on research and translation. Another thing is that I have limited time of my life. One day I will be old and pass away. I don't know whether I will able to kill a patient with free-living amoeba infections or not. But I know that all the knowledge we have obtained, I have learned, will pass to the next generations like you in the future. And hopefully, this disease will be killed. This is the biodesign motto. As a physician, one can help a limited number of people. As a physician innovator, one can change the world with a single innovation. And that motto is actually, this quote is in my statement of purpose when I applied for my PhD degree. And I'm still honest to myself that one day my inventions may be benefit a single or many people. And I give the credit to Professor John Boitano, my friends, and also my consultant for enlightening me to interest in doing research. I would say that you have to ask yourself why. Why you're doing research? You're doing it for yourself, or you're doing it for others? It's all start here from the bedside, from the quote, "There's nothing we can do." But I believe it will not never end here. Oh, sorry. And thank you very much.
Thank you, Associate Professor Dr. Pachalapon, for that exceptionally valuable session. Your insights have truly deepened our understanding of research and parasitic diseases.
We are now in the Q&A session. Since we are tight on schedule, we will only accept one question for this section. So for those interested in asking a question, please raise your hand now.
Okay. Thank you, Dr. Sara Sambat, for your nice presentation. I'm just wondering, while you are regarding the diagnosis of pneumocystis cerevisiae, so the staining procedure is available, some staining procedure, because PCR is not available in all stations. So in that case, some staining procedures are available or not?
Yes, from our study, we did all the staining, like GIMSA stain, GMS, and IFA. But there's a limitations of the sensitivity of that test. That's the reason why we want to come up with a more sensitive test. Because the patients who are non-HIV, sometimes their parasitic burden is low, but the immune is really weakened. That's the reasons why they develop PCP though, and we misdiagnose or underdiagnose this group of the patients though.
Okay, and one more question regarding the treatment actually. Sometimes you know that the sulfamethoxazole is quite effective. I'm just wondering why this antibacterial drug is much more effective than other drugs.
Right? Okay.
Bactrim is
I'm not
used,
really familiar with that though, but for the living amoeba, there's many components that you have to use though, since this the infection is really rapid. The disease progression is really rapid. The patients die really quickly. So we want the drug that have really high CNS penetrant, and also we have to actually modulate the informations that happen in the brain too. So for treatment, there might not be a single drug that you can use, but you have to find other modalities to treat this disease though. So that's why it's very hard.
Okay, thank you then.
You're welcome.
Thank you, Associate Professor Dr. Pachalapon, and to our participant. Your questions really help clarify the key points of the session. Before we move on, please allow us to present to you with a souvenir as a gesture of our gratitude.
As we come to the final session of this morning's program, we are pleased to welcome Professor Dr. Janjira Thaipadungpanit from the Faculty of Tropical Medicine, who will present genomic tracking of the human leptospirosis epidemic in Thailand and explore how modern techniques allow us to track and control infectious diseases. Please join us in welcoming her to the stage.
Thank you for the introduction. So I'm the last one, and I'm trying to be short but concise. I'm not here with the Siriraj Hospital, but I'm from the Faculty of Tropical Medicine, Mahidol University, in the Payathai campus. Thank you for the organizer to invite me here. It's my honor. I'm not a medical doctor, but I'm PhD student with work a lot with medical research. I have a good colleague, physician that help a lot on the medical research. I start my medical research during the PhD. I start with leptospirosis, and then since more than 30 years, I still, even though I moved to other disease, but I still keep leptospirosis as one of my focus. So today I would like to present you like one aspect of our study. This is more like a leptospirosis. It never leaves us, Thailand. It comes as a zoonosis, and then it peaks during the flooding time in northeast of Thailand. But nowadays, more cases in the south of Thailand as well. And you had heard before that leptospirosis is transmitted from rodent to human, or rodent to wild animal or farm animal, and then human exposed to that. But we just published that not only cat, also not only rat, but pet like cat and dog can transmit leptospira to their owner. So be careful. I will start with a severe case that we found in Bangkok, actually. This is Myanmar worker that work in the market, wet market. Let's say that they transfer the raw chicken in the market. And one day he had been bitten by a rat. Even though he wear boot, he didn't care so much, just drop some iodine. And then next, he developed fever. And then he have a calf pain, have really difficulty in movement. And then he come to a private hospital on like after 3 days, right here. And then they got some antibiotic for 3 days, but because of the too much cost on the private hospital, he had been transferred to our hospital at day 5. During that time, no lab had been confirmed that he got leptospirosis. My physician friend gave me the blood on the first day to do the PCR, and then we confirmed that he got a very low amount of bacteria in that blood sample. So we confirmed that they are leptospirosis. However, this is not official. Our lab is for research only. Then all the patient needs some confirmed case from the MAT, that is serology. For the MAT test, you need pair serum during the fever and after the fever, like 2 weeks. Then the MAT confirmed that he got really leptospirosis. So he is normally leptospirosis, only 10% of leptospirosis case is severe case. You got hemorrhagic fever, pulmonary hemorrhage, jaundice, and have a oliguria, like this. So in that time, I think the differentiate diagnosis quite difficult because of we can confuse with dengue and malaria. But these two disease, we have a rapid diagnosis that you can rule it out easily. Because of dengue, you have like NS1 rapid test. Malaria, you can do a thick smear, thin smear. But rickettsiosis and leptospirosis is quite difficult. Because of these two bacteria normally have very low load in the bloodstream. So to confirm, only this is only confirmed by serology. They have not, without PCR, you have no early diagnosis for treatment. And even though in your hospital have a PCR available, another difficulty is you need to send the blood during the narrow window period, 1 to 3 days after fever. Then if you are like a new physician and doesn't know that, okay, anybody can send, if you send the blood to test the IgG or IgM for leptospirosis in that day, it's not gonna work, and it might lead to misdiagnosis. So, or if you send the blood for the PCR, but patient have been sick for like 5 days, the chance of getting positive on that PCR is very low. And this paper, it shows how it's difficult for the disease to be misdiagnosed. Based on clinical suspected case, you can see that only 20% that had been confirmed leptospirosis by serology or by culture. So without PCR, so it's quite difficult to accurately diagnose. Then why I'm talking about it? I just would like to say that leptospirosis is easy to treat with doxycycline, but it's quite difficult to accurately diagnose. And another thing that the molecular microbiology found out is that leptospirosis didn't caused by only one genus. Caused by only one genus, but that genus have like 68 species. This is quite complex enough. At first, it's way more easier than this. First, they have only one pathogen called Leptospira interlegan and one non-pathogen called Leptospira bifexa. But after we learn and molecular era, we found that more than 8 species that can cause disease, human disease. And the other group here, we call it P2, can cause like mild disease. In Peru, they proved that our Leptospira wolfii can cause mild disease. Then there is another related to the really pathogen species. It's this group P1, that too. This is only found in environment specimen, sample, let's say like that, but no evidence on causing disease yet. But based on molecular, it's very, very close. It's just a time that we might see that there is some case that cause disease by these unreported species. So, however, since the species or thing is complicated, the patient and doctor still get familiar with Silova infection. They will ask, the MAT test, that is a confirmed test, and what is the Silova that caused disease? And not only that, the rapid test, IgG and IgM, sometimes they're based on the Silova. This table shows you that one Silova can be more than one species. Silova is more like a fashion clothes. They changed the clothes, but they didn't, the clothes doesn't reflect what they are inside, you know what I mean? So they can change the serotype that affect our immune as well. So if you have been infected by, let's say, bacteria before, and then bacteria could be interleukin and xantholocyte, then if the rapid test or MAT didn't have the bacteria to react with, then how to predict the Silova that infection is quite difficult, and this is complicate the vaccine as well. I will skip this. And then to show you the burden of the leptospirosis globally, the first burden is in Oceania, then it's up in Southeast Asia and Caribbean. And this is show the diversity of the species that cause leptospirosis worldwide. To be noticed, our interleukin is dominant in most of the continent compared to Bog-Petersenii. Only one area here that Bog-Petersenii is dominant compared to our interleukin. I will example of the epidemic of the leptospirosis, which we only see just one for the 30 years. Here, during nearly the 2000, the year of 2000, you can see this peak. This is the case report to our NIH, กรมควบคุมโรค. You can see that we starting from like a few hundred cases in 1990, and then it climbed up, and then it obviously observed during the 2000, and then this peak is going down. Right now, into about this moment, we never had case more than 6,000 cases a year ever. So this is still the puzzle for everyone. Why in Thailand in that period we got a huge leptospirosis case increase? Then that during my PhD, my boss said that, okay, Janjila, they have a MLST, and then I said, what is MLST? Oh, it's that it's a multi-locus sequence typing. It's a molecular tool that you need to develop and to solve the puzzle of the peak. We would like to know why the case is coming up. Is it from the single clone, or it means it from the one sort of outbreak or not? So, and then what is the MLST, if you ask? It's we sequence a short, like a 500 base pair of 7 genes. That 7 genes, we select that it's a housekeeping gene. It means that the gene that it will have, we make sure that that gene appear in every Leptospira genus. And then it's more like a telephone number. If the sequence of, let's say, gene A is similar of gene A of one strain, is similar for it the same of the other strain, then the profile is not changed. We call it allele. We call it the same allele, and then we give the number for that. Then it more become like a mobile number for each isolate. And then we can say that it's sequence type 1, 2, 3, and 4. Then after develop that at my thesis, the scheme it worked quite well. As I told you that this is Leptospira genus, so the MLST for the whole genus. Because all the isolate it come from patient in Udon Thani. Then all of these come from human. And then we can prove that the scheme can differentiate different species. You can see from different color and it's different bunch. And if you zoom in, you can see that if we do the MLST from to the reference strain that it comes from many places in the world, the tree can separate into like a country specific as well. Like a Philippines, it's sequence type in Philippines is cluster in here, and Australia is there, and Papua New Guinea and Australia. But they have many groups because of the difference of the isolate in that area. It's quite diverse. Then it proved that our tool it worked. Then I apply to the Udon Thani isolate first, and then some mouse, not mouse, bandicoot rat, it no puk na na. This bandicoot rat normally the farmer it trapped for eating as well in Kola, Nakhon Ratchasima. And we apply to the isolate that come from patient in other province, the other 8 province. We reconstruct this phylogenetic tree. You can see that all of the Udon Thani in red dot is very close to each other. It's exactly the same ST. It's called ST34. And the lat isolate from Nakhon Ratchasima, it's the same as ST as well. And the other, we found the other ST34 in the other province as well, even it in the south or in the north. So ST34, it seemed like distributed in the whole country. But it's not alone because of the other isolate, which are not ST34. We can find that ST49, 46, 27, 41, 37. This is ST that we found in Udon Thani as well. So it seems like in the population of the Leptospira that acquired by our patient, it's quite diverse, but among that diversity, we found really an outbreak that caused by ST34. So we publish on that. You can track that to that paper later if you're interested. But the question is not finished yet. We still think why the outbreak it emerged, and then why it's suddenly going down. What is special for this ST34? Why it fit? Why many people get infection? Then we dig deeper. We use the whole genome sequencing, and then at that time I learned at St. Andrew in Scotland and collaborate with the Wellcome Sanger Institute. I as a molecular microbiologist, then at that time it's still we do the short read with the Illumina. That is the first few years that my it launched, I think. Then we do the whole genome sequencing. We compare that genome, and I'm not sure you can see it well. But if you look at the whole picture, the ST34 is clustered in a very small dot here. And then you see like a very long branch of other ST that we found in the Thailand. So these seem like, oh, it really the outbreak because of the ST34 is very small dot that you can see. Then we have to zoom in, cut out the other ST that quite distant. And then you can see that in this chunk a bit, it's a ST34 clone. So all is ST34, but the isolate that in the Udon Thani, it grouped together here in V, or when we enlarge it in here. And you can see that in that, even in that, so this is more like in every patient in Udon Thani that we study, it's still separate into two groups, cluster 1 and cluster 2. I can enlarge that a bit here. So if you can look at the cluster 1, I call Udon cluster 1 and Udon cluster 2, you can see there are something else. Like one patient in Nakhon Ratchasima, it here, it cluster in that cluster. And then we can see Chumpon class, it's cluster in here as well. So, but not the Lampang or Uttaradit or Layong or Chonburi that put in the Udon Thani cluster. This is quite hint or suggest that the epidemic of leptospirosis at that time occur in northeast of Thailand might caused by one sort of the leptospira that distribute in the rodent or we don't know animal host, but we cannot answer why it distribute a lot like that. And then we got the isolate from Vientiane, Laos as well. And then we plot how many isolate that in the cluster Udon and 1 and 2 between Laos and Udon Thani, because you know that Laos and Udon Thani have a Khong River. Sometimes Khong River is very low, so human can cross over, or the rodent can cross over as well. Then I think that so far we can prove using the whole genome sequencing. And with that too, I mean both MLST and genome sequencing, right now it's online, and then all the data from Thailand and everyone in the world that use the same tool that used for typing or typing for epidemiology study. All the database is in the pubmlst.org, which can serve it for everyone in the world. And the paper, this is my student at that time. She do more than me, then she add more one species together. So that is the second paper from that. And then this is about 1,796 isolate that submit into the database. Not from Thailand, that you can see from the map that it from many countries, Brazil with a lot of leptospirosis epidemic as well, and Philippines, yeah. So I come to my conclusion. From this, I think you can see that sometimes molecular, you can see for many examples from Ajahn before that molecular genomic sequencing can help in many ways, like diagnosis design or targeted therapy. But for me, to know the epidemiology, to know what your enemy are or can transmit to our human, then we can manage or we can protect or even we can live better with them. I think that genome sequencing or molecular tool is a tool that we need to more like keep at a data for further and further development or improving our disease management. Thank you.
Thank you, Professor Dr. Tantila, for the engaging presentation. We will now open the floor for questions. Please raise your hand if you'd like to ask anything. Mic here, please.
Thank you, Dr. Janjira, for your nice presentation. I'm interested about this ST34 clone. This was isolated from some animals also from the same time? Or only from the human samples?
Or it from human sample. We have only 5.
Okay.
5 from bandicoot rat. And all that 5 bandicoot rat, we got only 2 that ST34. Right. Okay. But we we got the DNA from rat in the different province. Okay. But is there evidence for the carrying in the rodent? Yeah.
As we know that this organism, Leptospira interrogans, it can be isolated from other animals also. So here it was only isolated from rats, is it? Not from cattle or other animals?
No, not yet. Because of that time, they have many paper. The other paper, they trap a lot of rat in Thailand, in many provinces. They found many sequence type, as we found in that 5 rat. Not dominant with ST34, right. like in the epidemic we found in Udon Thani patient. Yeah. Okay.
One more curiosity, actually, regarding the microscopy for the diagnosis. Did you try for the dark ground microscopy for the diagnosis?
Yes. Normally, we not directly look at the leptospira in the blood specimen. Normally, we culture first, or the isolate that we got from culture from blood. And then every week after inoculate the 1 drop of blood into the EMJS media, we have to observe under microscope every week until 1 to 3 months to make sure that it will not become positive. Then we have to observe under the microscope at that time. Because of at that time, or even now, to do the whole genome sequencing, we need to got the culture first.
Okay, thank you then.
Are there any more questions? If not, thank you for your thought-provoking question. We would like to extend our heartfelt thanks to Dr. Tanjira for her thoughtful responses and for enriching the discussion.
We'd now like to present you with a small gift to express our sincere appreciation.
This brings us to the end of this morning's conference. We hope that this morning's sessions have offered valuable insights and deepened your understanding of medicine.
And for our high school students today, we hope this experience has sparked the curiosity in each of you and encouraged you to continue exploring in the field of medicine and research.
If you have any further questions to the speakers or for any collaboration opportunities, please approach our staff or leave your contact information at the front of the stage after the end of this conference. Before we conclude, we would like to invite everyone for a group photo.
Could everyone please move into the center of the room for the photo?
We'd like to ask everyone to stand up and then move into the center of the room if possible. Thank you very much. Okay, now we will take our photo. 3, 2, 1. If everyone could raise their index finger for Siriraj 1. Thank you so much to all of our participants. The pictures will be available on Facebook.
That was lovely. With that, we formally conclude our conference. Thank you all for your presence and active participation. It has truly made this session meaningful and engaging. We hope you leave with valuable insights and inspiration, and we look forward to welcoming you again at future events.
Before you leave, we kindly ask you to scan the QR code displayed on the screen above to complete our feedback form. Your input is greatly appreciated. We wish you a pleasant day ahead and safe travels home. สวัสดีค่ะ
Please wait a moment while the QR code is being displayed.